You really did.
Someone might say you might go, yeah.
Got your coins?
I don't think she
All
likes.
right. I love how everybody always waves at this. Like it's the most exciting part of your day.
Can't see myself leaving.
All right, while y'all are getting your lunch out and getting yourself ready, excuse me for a minute. I'm gonna go take my contacts out because my left one is bothering me, and I'm gonna wear glasses, which y'all have never seen, so don't laugh at me. But I'll be right. I'll be back in just a minute. It's just making me crazy, and I'm like, I don't want to keep scratching out my eyes with every button. I'll be right back, okay? Get your lunch out, get your notes out, and get it.
No, I just like. I think it's like yeah. That's what it was. What? This is 39, dude. You see? I like booty. What? What do? Why are there so many young? Also, like added
I don't know if anybody else is super nearsighted, but I have this weird thing where I have to adapt to my glasses because they make everything smaller so that I can see it. So when I put them on, I feel 12 feet tall and I have to walk down the stairs looking straight down because I can't see. And then I do the weirdest thing. And this is the weirdest thing. Tell me if I'm the only one that does this. Actually, I hope I'm not because that's just so strange. But if I'm fixing myself a plate when I'm wearing my glasses, I put too much on it because everything looks smaller. So I inevitably like pile on the mashed potatoes and whatever I'm eating. And then I'm like, why can't I finish my plate? Because I put enough on there for a giant and I'm not that hungry. I just, everything's small.
Never eat directly after.
Like, I'm like legit brief line. So anyway, that left contact was being a little, you know what. All right. Thank you for your patience. Now I'm dizzy because everything's weird, but I can see. All right, so today is photosynthesis. Before we start, I should have given you this before I went upstairs. A couple things. First, you're going to need a sheet of paper at one point today. So I'll pass those around if you don't mind. And here's some for you guys. And here's some for this end. Because we're going to do an interesting note taker thing. Pass them around. And then I'm also going to give you, right now, before we start, your Q2 packet. So you have your binder. Open it to where it says Q2, which is one of your little orange pages at the back. It's going behind the one that says Q2. Sometimes when I do these, I like print them all out and then I hole punch the whole packet at one time. And sometimes I hole punch like all the lesson 12 and all the, thank you. You know what I mean? This time I did it that way. So you may have to fuss a little to get it into your binder neatly. But I'm going to bring them around, make sure everybody gets one. Nobody's absent, right? I don't see anybody missing. All right. Huh?
I'm absent.
You're absent?
My good time like you, a teacher said raise your hand.
That's a good one. No.
Wait.
But you got yours, I was trying to give it to you, forgot. All right, can you pass that to Claire, because I can't quite reach her. And then this one's you, really?
Get back.
You.
Why not?
You.
Why not? Yeah.
All right, this is you. So to the right where he is, in the back, you'll take YouTube, and Q3.
Those two things. It's YouTube. You do those.
Sure, has this race space. And then that's, yeah, excellent, yay, have one extra, that's what I'm supposed to have, so. All right, there we go, good deal. So now you've got Q2. Hooray. I try to get him to you a little early. Not like the day before.
Yeah.
Yeah. Excellent. All right. So your homework talked about organization of life from atoms to molecules and compounds, although molecules is always going to be appropriate because we are talking about life, so it's organic, to cells, to tissues, to organs, to organ systems, to an organism. Now, couple of you did the right order, but you got it backwards. You put atoms where organism is and vice versa. But the idea of a diagram like this shows that organisms contain, because it's the bigger circle, organ systems, organs, tissues, cells, and such what, right? But atoms only contain themselves. So that's why you have the smaller circle going to the bigger circle, right? Excellent. All right, so this is just another example of models. We use models all the time. This is a good one. All right, now let's see if we can figure out what goes here. First off, what kind of a cell is this? A plant or an animal cell over here on the left? Barrett.
That's a animal cell.
That is an animal cell, so the other side is a plant cell. Very good. What is this that's only in the plant cell? Drew? Well, it's this one right here. This top one's pointing right here. Yeah, right here. No, because it's only in the plant cell. The cell wall is right here, and you're correct. That's also in the plant cell, but get rid, who's that, Eunice?
Is the Golgi apparatus?
No, it's not Golgi apparatus. Golgi apparatus is in both cells. Cullen. Chloroplasts, there you go. So your chloroplasts are right here. You're often going to see them look like that. They're going to show them like that frequently. A blob with little circles in it. That's how we model that. And then Drew, this would be your cell wall because that's pointing right there. So the next thing is in both cells, right here and right here. What are we looking at, Barrett?
Cell membrane.
There's your cell membrane. That's going to be in both. Very good. Now it's pointing to the, excuse me, the little dots. What are the little dots? They're in both cells. Eunice?
Nucleolus.
No, not there. Right here. See, it's pointing this one right here. It's pointing right here. And it's pointing right here. Barrett?
Cytoskeleton.
No, this is going to be your ribosomes. But good guess. All right. What's the next thing it's pointing to? It's in both plant and animal cells. It's this guy right here. It's got squiggly lines in it, not circles. Barrett?
Mitochondria.
That's a mitochondria. Very good. Sometimes you'll see it instead of a squiggly lines with like a ziggy-zaggy line. That's often how it's represented. All right, now what's this thing right here? The squashy looking looks like a splat. It's in both of them. Anybody? Barrett again?
Those the endoplasmic reticulum.
Nope. Close though. What you got?
Vacuole.
Not a vacuole. Last week I said some people say it looks like a stack of pancakes. Kenna. Golgi apparatus or Golgi body. Very good. That's what that is. Very, very good. All right. Now this is attached right on the outside of the nucleus. And you notice there's two things on the outside of the nucleus. There's this guy here, same on both sides. And then there's this one. So we're looking at this one here and here. It's got dots on it. What is that? You know?
That's the endoplasmic.
Which kind? We've got 2 endoplasmic reticulum.
What are the names of the two?
So you have a rough and a smooth.
Those are rough.
That's the rough. Very good. Okay. And the reason why is because the little dots represent your ribosomes, which remember were these little guys out here. So that's where your ribosomes are produced, or on your rough endoplasmic reticulum, and then there are little dots everywhere else. So if that's the rough, then what's this one, everybody? Smooth endoplasmic reticulum. All right. The next thing it's pointing to is this line right here and right here. It makes these little circle. This line, what's the line going to be, Caitlin? You lost it? You tried? Anybody? So that's going to be your nuclear membrane. See, that's the membrane around the nucleus and around the nucleus. And then what's it pointing to here? That's the whole big circle. What you got? That's your nucleus. Very good. And then inside of your nucleus, it's pointing right here to this little dot. What's that? Nucleolus. Very good. And then what's the last thing we're pointing to? Over here in the plant cell, it's really big. And in the animal cell, it's smaller. What is that? Jordan? Vacuoles. Very good. All right. So we're getting there. We're getting there. Now, you'll see there are like a million different cell models that you can go with, right? But a lot of them are similarly represented. The organelles are similarly represented. Like chloroplasts are frequently going to look like blobs with little circles in them. Ribosomes are frequently going to look like little dots. So it's just kind of how they're represented. And you'll see it again and again when you see more representations or models of cells. All right, so let's talk about energy in life. All right, what is adenosine triphosphate? All right, CJ, what you got?
It's the chemical compound cells use to store and release energy.
There you go. So this is ATP. It's your primary energy carrier in all living organisms on Earth. Microorganisms capture and store energy metabolized from food and light sources in the form of ATP. You have ATP. There's a whole big, we're going to do a whole big thing with ATP in just a little bit. That's what your little piece of paper is for. It's a whole, we're going to model photosynthesis physically, and you'll see what I mean. All right. So ATP is the end product of the process of phosphorylation, which is adding a phosphate group to what? What do we add a phosphate group to, Yusuf?
Adenosine diphosphate.
Adenosine diphosphate. When you add a phosphate to adenosine diphosphate, you get adenosine triphosphate, which is ATP. Okay, ATP is, remember this one forever. ATP is your energy molecule. It's what you need, right? It's the forming and breaking of it is essential in energy production and storage. So cells release energy by the removal of a phosphate group, which converts what to what? We just said it.
ADP.
If we're removing one, we're going from ATP to ADP. All right, very good. So all living things use ATP. All living things use ATP. In addition to being used as an energy source, it's also a signal transduction pathway for cell communication. It's incorporated into your DNA, and during DNA synthesis, cells regenerate ATP from ADP that only contains a small amount of ATP at any given time. ATP is fleeting, okay? ADP sticks around a bit. ATP, the production of ATP is a fleeting thing. You add the phosphate, you remove the phosphate, there's an energy transfer that happens there. And that is essential in our production, our metabolism of energy, all of that. So other molecules are related to ATP and have similar names such as ADP and AMP, which would be adenosine what? Monophosphate, adenosine diphosphate, adenosine monophosphate, adenosine triphosphate. How many phosphates do you have? It's right there in the prefix. Mono, tri, di, right? We all know mono, di, tri, right? Okay. So in order to avoid confusion, it's important to know the differences between them. So we have heterotrophs and autotrophs. We've talked about this. So what uses inorganic energy sources such as sunlight as a source of energy? An autotroph or a heterotroph, Drew? An autotroph, okay. And then what's going to use organic sources such as other living things? Asher, a heterotroph, right? Auto, self, hetero, other. So what are some examples of autotrophs? Somebody give me an example of an autotroph. Xander. An autotroph. A plant? Give me a plant.
All of my sunflower.
A sunflower. Okay. How about another one? Autotrophs. A tree. Give me a tree.
Oak tree.
Oak tree. Okay. Give me one more autotroph, somebody.
Algae.
A what?
Algae.
Algae. That's a great one. Algae. Yep. Okay. And give me an example of a heterotroph. Lucas.
Me.
You. Jordan.
Here.
A deer. Very good. Give me one more. Tyler. Can you give me one? A heterotroph.
Human.
Hey, I'll take it. You get a coin. All right, there we go. Awesome. Very good. All right. And photosynthesis is your process that uses light to produce food molecules. We all know this, right? You've known this since you were tiny. But what you may not have known is how it works. So we're going to talk about how it works. All right. So you have chloroplast. Now do you see why that chloroplast is represented by a blob with a bunch of little circles in it? You can see the little circles now, right? Okay, so what are your light absorbing molecules that capture the sun's energy? Those are your pigments in general. Pigments in general, okay? And what is the pigment that absorbs all wavelengths of light except green, which it reflects? Caitlin.
Chlorophyll.
Chlorophyll. Okay. If this is orange, it's absorbing every wavelength except this particular shade of orange, right? This is absorbing, well, it's partial, it's translucent. The green is reflecting the green, right? So it's absorbing everything except that color. Caitlin. Who is?
Mirrors.
Mirrors are green? How do you mean?
When you see the side of it, it's green.
Sometimes, yeah. So that's like here. And that's because of whatever's in the this particular glass, whatever impurities are in, well, or mixed in intentionally with the silica. Like if you ever look at if you look through your car window, it looks pretty clear. But if you've ever seen a car that's been in an accident, the glass tends to have kind of a blue or bluish green hue. And that's just they've added other chemicals to the glass as part of the manufacturing process for various reasons to get different results. So yes, chlorophyll is your green pigment. So it's reflecting the green, it's absorbing everything else. So what would this be? Each of a number of flattened discs, oh, looky here, inside a chloroplast bounded by pigment and membranes on which the light reactions of photosynthesis take place and arranged in stacks or grana. What are these little guys called? These little discs in here?
Thylakoids.
Thylakoids. Okay, these are called thylakoids. They have a little membrane around them. And that membrane around them is where you've got your photosynthesis taking place. And they're in stacks, and we call the stacks grana. They look like some people will say they look like a stack of coins. And they really do. They really do. So that's your thylakoid. And then what's the fluid matrix that surrounds, look at that weird thing, the T didn't move, surrounds the thylakoid and the grana? Yusuf.
Stroma.
Stroma. Okay. So that's what we call this little fluid matrix that's just floating around in here. All right, so we have these little stacks of green coins. They're thylakoids. They're inside the chloroplast, and the actual photosynthesis is taking place on the membranes of these little guys. So I love this video. I mentioned it last week. It's really cool, but it takes you on like a little journey into a chloroplast, and you can actually see what's happening in there and the structure up close. It's pretty cool. Why does it want to do that? Go back to the beginning. Okay, play it. That's a stomata. That's an opening in a leaf. And really, there wouldn't be this much matrix. They'd be kind of squished together and they'd be more boxy, but it's the best they can do graphic wise, I guess. You're inside the cell. Now you need to go inside the chloroplast. Always looks like a sci-fi movie to me, but it's so cool. So those are all the molecules undergoing their different chemical reactions. All on the membrane of the thylakoid, inside the chloroplast, inside the cell, inside the plant. So you went deeper and deeper in and you could actually see kind of where everything is taking place. Okay, so we have what we call light independent reactions and light dependent reactions. Now, the light independent reactions, you will sometimes, especially in older textbooks or older videos, you will hear them referred to as dark reactions. And I know that confused me the first time I heard it because I was like, oh, do they only take place in the dark? No. They just don't need light. So it's better to call them light independent. That's much more clear. But if you ever hear the term dark reaction, just know that's what we're talking about. So what are your functional units for photosynthesis, which are defined by particular pigment organization association patterns? What are they? Photosystems. Okay. So these are functional units for photosynthesis defined by particular pigment organization and association patterns whose work is in the absorption and transfer of light energy, which implies transfer of electrons. Physically, photosystems are bound in the thylakoid membrane. So all that little molecular movement you saw, as a whole, we call that a photosystem. So there's a lot of electron movement. And if you remember, like from those of you that did chemistry, you know, when you get electrons moving, you have energy either released or absorbed. That's where you're getting the energy transfer here for, yes. What? Oh, the next one? Okay, what's the next one?
Like electron transport.
Electron transport chain. So the electron transport chain, very good, is a series of electron transporters embedded in the inner mitochondrial membrane that shuttles electrons from NADH to FADH2, these are all molecules, right, to molecular oxygen. In the process, protons are pumped from the mitochondrial matrix to the inner membrane space, and oxygen is reduced to form water. What does it mean to be reduced? Who took chemistry? What's reduced? Reduced. Remember, we had oil rig. What is reduced?
Getting like it's the opposite of oxidization.
Yeah. Remember, oil rig, oxidation involves
Loss.
loss. Reduction involves of what?
A gain of oxygen.
Not gain of oxygen.
Electron.
Electrons. Thank you. Yes. So if we are reducing a substance, it means we're gaining electrons. I know that sounds counterintuitive to those of you who haven't done chemistry yet, but that's what reduction means. Okay. So what's the protein that catalyzes the formation? What you got? ATP synthase is a protein that catalyzes the formation of the energy storage molecule ATP using ADP, an inorganic phosphate. Phosphate's not organic, right? It's phosphate. And it is classified under ligases, which is just a type of molecule. That's what we're calling these, as it changes ATP by the formation of a PO So that's a phosphate oxygen bond, phosphodiester bond, we would call it. So this where you get, this is your organic chemistry. Remember I told you organic chemistry gets real fun because the vocabulary is like, off the wall. Oh man. So it's going to make more sense in a minute. All right. So light independent reaction. So what's the process that plants and algae use to turn carbon dioxide from the air into sugar? Okay, Eunice.
Calvin cycle.
The Calvin cycle. Yep. Okay. It turns carbon dioxide from the air into sugar and the food autotrophs need to grow. Every living thing on earth depends on the Calvin cycle. Okay. I'm going to make this point right here. I get really annoyed when people say, I don't eat any sugar in my diet. Well, you're going to die. Enjoy it because you require sugar to function. And people say, I don't have any sugar. I'll just eat this apple. I don't eat sugar. I'm going to eat celery or salad or anything. Okay, anything. There is sugar in nearly everything you consume. What people mean when they say that is I'm not adding sugar. I'm not adding cane sugar or high fructose corn syrup or any of this other stuff. That's what they mean. Note that your body fundamentally is going to take all of the different sugars it consumes, fructose, for example, high fructose corn syrup. It's going to take all of that and convert it into glucose. That's what it's going to do. Sucrose on the table, pour that in your cereal in the morning or whatever, it's going to convert it to glucose. Okay, that's the sugar we use. When you eat an apple, you're taking one sugar, you're converting it to a different kind of sugar within your body, but sugar isn't everything. You have to have it. It's essential. Like you cannot live without it. The people that say, I don't need any sugar, I'm like, every time, like, okay, you're still walking around somehow. That's amazing. So the Calvin cycle has 4 main steps. Carbon fixation, a reduction phase, which means we are gaining electrons, right? And a carbohydrate formation, and then a regeneration phase because it's a cycle. It goes round and round and round and round and round. Here's the thing with these cycles. For the most part, I'll let you know if it's otherwise before tests or exams. For the most part, I want you to understand it less than I want you to memorize it. There is never a point in your adult life, unless you go into botany or you go into some biological or medical field where somebody's going to say, tell me how the Calvin cycle works and give me all of the molecular steps involved in all the chemical reactions. It's not a thing. But what you should understand is how it works and where it's important. Okay, so energy to fuel chemical reactions in this sugar generating process is provided by ATP and NADPH. It's 2 different chemicals that work in your, I'm working this whole process here. You'll see them in a bit. Chemical compounds that contain energy, the energy plants have captured from sunlight. So these are energy compounds. So here's your Calvin cycle. It's what kind of reaction? Light dependent? What'd you think?
Light.
So it's one of these, this is what we call dark reaction, which really means it's light independent. Okay, so it doesn't need sunlight to happen, although it can happen during the day. So it doesn't require the energy from the sun to work.
Why do we see the light coming into it?
Well, because you need the light reaction in order for the light, you need the light dependent reaction to occur in order for the light independent reaction to occur. Because the light dependent reaction,
Something like spider.
yeah, I just saw, I can't see stuff out of my, no, I think it was a piece of lint. I don't know. But I can't see anything in my periphery. It was like something's moving. My glasses on, it weirds me out. So you need the products of the light reaction in order to do the light independent reaction. So that's why you see the light coming in there. So this is your light reaction right here at the top, or your light dependent reaction. And then this is your Calvin cycle down here. So you can see your light reaction is putting ATP and NADPH into the cycle down here where you have your light independent reaction. All right, so here's another quick one. There've been some pretty decent graphics on this. These are a little older. They might be better ones nowadays, but I've always liked these.
Plants provide us with food to eat
Really?
and oxygen to breathe.
Thanks.
They perform this amazing feat by the process of photosynthesis. Let's take a closer look.
Okay.
Photosynthesis requires carbon dioxide, which diffuses into the leaf through small pores and then enters the cells. Inside the cell, carbon dioxide diffuses into the chloroplasts, where photosynthesis takes place. Chloroplasts use energy from light to transform carbon dioxide and water into sugar and oxygen. Zooming into a chloroplast, we see these flattened membranous sacs called thylakoids. Here, light energy is converted to chemical energy in the first phase of photosynthesis, the light reactions. The two green structures you see here are photosystems, large complexes of proteins and chlorophyll that capture light energy. An electron transport chain connects the two photosystems. Notice the small mobile electron carriers that shuttle electrons from one large
The little guy
complex
going back
to
and
another.
forth in the middle.
Now let's take a closer look at the steps of the light reactions. The photosystem on the left absorbs light energy, exciting electrons that enter the electron transport chain. Electrons are replaced with electrons stripped from water, creating oxygen as a byproduct. The energized electrons flow down the electron transport chain, releasing energy that is used to pump hydrogen ions, the blue balls, into the thylakoid. In the photosystem on the right, light energy excites electrons, and this time the electrons are captured by an electron carrier molecule, NADPH. The high concentration of hydrogen ions inside the thylakoid powers ATP synthase, producing ATPs. The light reactions in the thylakoid have produced two energy products, ATP and NADPH, that will now power the production of sugar in the Calvin cycle. The Calvin cycle takes place outside the thylakoids in the stroma, the thick fluid of the chloroplast. At the beginning of the cycle, carbon dioxide molecules combine with molecules called RUBP. The resulting molecules go through a series of reactions powered by ATP and NADPH from the light reactions. Sugar molecules known as G3Ps are produced. Most of the G3Ps are rearranged back into RUBPs that will begin the Calvin cycle again. But the important product of photosynthesis is the remaining G3P sugar. Some G3Ps are used to build glucose, which can combine into starch or cellulose. Still other G3Ps form sucrose. And some of the sugar is broken down by cellular respiration using oxygen in the plant's own mitochondria, generating ATPs that can power other work of the plant. Excess oxygen diffuses out of the leaf through the pores, while more carbon dioxide enters. With three simple ingredients, carbon dioxide, water, and light, plants produce sugar and oxygen by photosynthesis, powering plant metabolism and ultimately providing your fuel as well.
So you've heard plants breathe out oxygen. Where did the oxygen come from? What was broken down to produce the oxygen? Huh? Not the carbon dioxide.
Water.
Water. Because what did it need from the water molecule?
The oxygen.
It needed, no, it let go of the oxygen. It needed an electron. And where's the electron coming from? Hydrogen. How many electrons does hydrogen have? One electron. So you break out that water, you get one electron. That's what it needs. You've added an electron, which is, you've gained an electron. What kind of reaction is that? Reduction. There we go. Okay. All right. All right. Complicated, right? Very complicated. Don't worry. We're gonna come up with a good, we're gonna have a good mnemonic here, a good memory device. So photosystem 2, from your notes, can anybody summarize? Eunice, what have you got?
Oxygen, protons and electrons. Oxygen is released.
Okay, so photosystem 2 absorbs light and increases the electron's energy level. The electrons are passed to the electron transport chain. Enzymes in the thylakoid break up water molecules into 2 electrons, 2 H+ ions, and 1 oxygen atom. Look, 2 electrons, that's coming from your hydrogen, right? How many neutrons does hydrogen have?
One or?
How many neutrons? Hydrogen. Come on, somebody who had me here last year. 0, 0, right? It's only going to give you, look, 2 H plus ions. It gives you 2 electrons and 2 H plus. So it's giving you 2 electrons and 2 protons. That's the H plus ion, right? And 1 oxygen atom. The 2 electrons replace the high energy electrons that have been lost to the electron transport chain. Okay, who can tell me about the electron transport chain from your notes? Should be there. Kenna. Okay, so yes, energy from electrons is used by proteins in the chain to pump H plus ions, as those proteins, right, from the stroma into the, protons, I can't speak, into the thylakoid space. At the end of the electron transport chain, the electrons themselves pass to photosystem 1. Honest to goodness, electrons are moving physically within these cells down on these little membranes, these thylakoid membranes. You have actual physical movement of electrons. That's how, anytime you're moving an electron from place to place to place, it's going to take energy, but if you're adding electrons or taking away electrons, it's all about energy transfer. Electrons moving around is what does that. Photosystem 1, who's got it? From your notes, from your notes, from your notes. Jordan, what you got?
It takes light to re-energize or re-excite electrons.
There you go. So the electrons do not contain as much energy as they used to. Pigments use energy from light to re-energize the electrons. And at the end of a short second electron transport chain, NADP molecules in the stroma pick up the high energy electrons along with the H plus ions, which are proteins, right? Protons, I keep saying that, at the outer surface of the thylakoid membrane, and that becomes NADPH. Okay, and then how about this last one? Hydrogen ion movement and ATP formation. Ken again.
Space fills up with positively charged hydrogen ions. The inside of the thylakoid membrane becomes positively charged relative to the outside of the membrane.
Good. So your hydrogen ions began to accumulate within the thylakoid space, and the buildup of hydrogen ions makes the stroma negatively charged relative to the space within the thylakoids. That's a gradient, a charge gradient, right? And the difference in both charge and H plus ion concentration across the membrane provide the energy to make the ATP. Okay, this is like what? I know, it's a lot. So let's talk for a minute. Let's kind of break away from the whole nitty-gritty process in the weeds, back up a little bit, and let's talk about the term photosynthesis. So this was coined by this dude in 1893 named Charles Reed Barnes, and he derived it from Greek words. And you can tell what those Greek words are, right? Photo means to synthesize, means make or build. Yeah, so building or making, creating from light. All right. Photosynthesis, light synthesis. Now this guy, Jan Ingenhose, understood the process in the late 18th century, noting that he noticed oxygen production in plants under light. So he's like, oh, something's going on here chemically because they are producing oxygen, right? Right. Okay. So we had a guy named Priestley. Good name. I like that name. Joseph Priestley. Because Jason Priestley was one of the guys that was on everybody's bedroom wall when I was a kid. If you don't know who he is, look him up. Anyway, this is Joseph Priestley. Unfortunately, it's not Jason Priestley. Anyway, conducted an early experiment on gases that led to the discovery of photosynthesis in the 18th century. And the role of light, Jan Ingenhose demonstrated in the 18th century that light is essential for the photosynthetic process, right? And then chlorophyll and oxygen, we learned in the 19th century from Julius Sachs, who discovered the role of chlorophyll and the production of oxygen in photosynthesis. So there's been a lot of people over time who have contributed to this. So this is what Priestley did. He set up a mint plant, at least he picked one that smells good, in a closed space with a burning candle to observe oxygen depletion and replenishment. Oh, that's smart, right? Because you need oxygen for a candle to burn. So what he found was the candle extinguished when oxygen was depleted, showing the plant's role in oxygen production. And he noticed that after some time, the candle could be relit, which demonstrated that the plant replenished the oxygen in the closed space. Really simple. That's pretty basic. I mean, that's pretty basic, but somehow ingenious at the same time, right? So it's the foundation for photosynthesis. The experiment laid the foundation for understanding photosynthesis and the role of plants in producing oxygen. Oh, there's our guy. Hello, Joseph.
Yeah.
He looks a little stuffy, but and they always look like, when they have those high collars, that just looks so uncomfortable to me. I can't wear turtlenecks. I feel like I'm dying. So like, I don't know how people did this on the regular. Yeah. Turn him into a rat. I probably should. I've turned a great many scientists into a rat. So what did Ingenhousen do? Ingenhousen discovered that plants produce oxygen bubbles on their leaves when exposed to the sunlight. And the experiment demonstrated that sunlight is essential for photosynthesis to occur in plants. And he concluded that plants undergo respiration similar to animals when they are in darkness. So even at night, plants are exhaling, right, oxygen. So he realized that. Oh, this is Ingenhousen. Yeah, he's got the little powdered wig deal. Oh yeah, gotta love the powdered wigs. It's funny, I was asking somebody the other day, like in some countries, their judges and lawyers still wear the powdered wigs. It's like part of their thing. Like, I think they still do it in parts of Canada. They do it in the UK. Like, it's the uniform. Like, you're going to court, you put on your white wig with your little, it's interesting to me that that's a thing. Like, it's kind of weird. Yeah, exactly. That's why they wore all the wigs, right? Everybody knows that the reason historically the white powdered wigs became a thing was because syphilis was an epidemic. People's hair was falling out. The moral past, everybody was so moral and ethical. Yeah, well, sometime this year we may touch on that because we will get into all the fun stuff. Okay, discovery of chlorophyll's role. Okay. So chlorophyll in photosynthesis, this is Sachs. So Julius Sachs discovered that chlorophyll is essential for photosynthesis, converting light energy into chemical energy. And how did he do that? So he demonstrated that plants produce starch only in the presence of light, emphasizing the role of chlorophyll. Pretty smart too. So the importance of light, he said plants need light for photosynthesis, highlighting chlorophyll's role in capturing light energy for the process. Anybody ever left something on your grass, like a board or a bag or a tool or your trash bin, and seen what happens to the grass beneath it within like 24 hours? Anybody ever seen it? What happened?
Brown.
Yeah, it turns brown or yellow. Yeah, because the chlorophyll is no longer needed there because there's no light getting to it. It's not going to produce any energy. So the plant's going to be like, well, oh well. You can actually, and it's kind of fun, but it's an interesting experiment. If you take something that's fairly opaque, like a poker chip or a coin, if you find a lightweight one, a poker chip, I suggest because coins are kind of heavy, but something like that's opaque, you can paper clip it to a leaf on a live plant, come back 24 hours later and pull it off. And depending on the plant, the species of plant, you may or may not see a color change in that leaf. It's kind of fun. You can do that. You can also use like black paper, but you may have to double or triple it up because you want it to be really thick. But it'll leave it's almost like an impression or a leaf tattoo of some kind practically. It's kind of cool. Yeah. Oh, there's Sachs. Some of these guys, who's the scariest one? Does anyone remember who I think the scariest looking historical scientist was?
Oh, I forgot.
It's the one with the eyes that looks like he's going to kill you. Do you remember who it is?
Oh, wait. No.
No, you're close. No, yes, it was Mendeleev. Sorry, I heard Mendel in my head. Yeah, Mendeleev. Dmitry Mendeleev. Yes, the guy who developed the modern periodic table is like any photo of him. You're like, please don't run into him in a dark alley. He looks so scary. This guy doesn't look so scary, but Mendeleev really did.
About making rat versions of them.
I need to, but I already had these made. Like all those rat ones were new versions, but I do need to go back and make rat versions of them. I have made AI rat versions of many scientists in the past, and they're quite entertaining. But he's
So
he looks like he's missing a pipe. Like he needs a pipe. There's a composer we come across every so often in choir. He's a modern day contemporary guy, but it's so funny because sometimes with modern day composers, they'll put their photo on the back of their music along with a little bio. And every photo of him, he's wearing a turtleneck, like a blazer with suede elbow patches, and he has a pipe in his hand. Every time I see him, I said, "This guy again with the pipe, holding it and posing for his picture." Yeah. So again, this guy Charles Barnes coined the term. So he's an American botanist, 1893. Charles Reed Burns coined the term photosynthesis to describe this biological process, and it's the process by which green plants use sunlight, carbon dioxide to create food, we know that, and water, and unifying biological understanding. So Barnes' term helped unify scientific understanding of the fundamental process. So good. Sometimes we need common terms. We'll talk about this when we get to taxonomy and naming species. But that's the great thing about our Linnean taxonomic system is we scientifically, we will call a species by its scientific name, which is its genus and species. So you'll name something as, whatever. And when you do that, that is universal because there are a million common names for things. Like sometimes you'll see somebody in the yard, like there's a particular weed that grows around here a lot. And the stalks are kind of magenta colored and it produces these berries that are like purple and they stain really bad. Some people call it pokeberry, some people call it inkberry. If you call it by its scientific name, everybody knows what it is, right? So that scientific names are great. And that's why we need definitions. This guy looks cool, man. I wanna have a cup of tea with him.
It's like a certain historical.
He's very spiffy. I like to see it. It's American. 1893. All right, now we're going to use another teacher's resource to review the process of photosynthesis. And you're probably like, why are we watching another teacher's review? Well, I'm gonna tell you. This lady's named Mrs. Sloan, Winnie Sloan, okay? And she teaches honors AP and I think on level biology. I think she's in California, if I'm not wrong. But her reviews and her modeling of processes with memory devices is the best I've ever seen anywhere. There's no point in reinventing the wheel. Hers are so good. She has an entire YouTube channel full of this stuff, and it's amazing. So the paper that I gave you before, get out that paper and a pencil. We're going to take some notes first. Yes.
You know the channel name?
I think it's Miss Sloan Biology or Winnie Sloan. I'll be sure it'll be included in your resources.
Yeah.
I have no idea, but she's great. She's really, really good. She's really good. All right, everybody's got their paper out. Now this is good practice, okay? Sometimes when you get into college classes or other classes, you have to take notes actively during class. So that's what we're gonna do. You're gonna be modeling some things out on paper, and then you're gonna model it with your hands. Okay, so I'll show you what I mean. All right, so we're going to watch her and then we're going to kind of model what she did on paper and then we're going to model it again. So this is her big review of photosynthesis and then she has like a condensed version. All right, now this is amazing. If you're somebody that struggles to remember all the steps in a process because they don't mean anything, this is a great way to do it, is to combine movement and make it almost like a chant or a song, which is what she does. Yeah.
Why does it say TikTok and then say what?
She has a TikTok version.
Why does it what?
Well, because this is on her YouTube channel, but she also has a TikTok and an Instagram and whatever else, and she's great. So this is the long version, okay? Then she has a condensed. So I'm going to show you the long one for context. Then we're going to model it out on paper, and then we're going to go back and look at the condensed, and it's going to make more sense. So just watch her. She's so cool. I love this lady.
We thought we'd invite you on it. So remember, we're gonna do photosynthesis within the form of.
Hold on. Alexa, turn on media center.
Why is the blood located where it is?
Oh, that's me watching my forensic stuff. It was really good last night, actually. It was very interesting. Okay. Now No, it was fascinating because what it was, the first one, it was called some forensic show in Peacock, but the first one was about accident reconstruction. So it was all about angles, math. It was about a pipe that a couple was driving down the road, and the man claimed that a pipe fell off a truck, came through the windshield, and whacked his wife in the head, and she died. And they were like, "Nah, you killed her, and here's how we can prove that pipe didn't come through the window." It was all about the math. It was so cool. And then that one was about other forensics. Anyway.
What happened?
Yeah, and it wasn't broken in one place. They saw two bang, bang, so they could prove it didn't just fly through. It was stuff like that. It was really cool. Anyway, I like stuff like that. Make science super useful and applicable. All right, let's try Miss Sloan
So
again.
remember, we're gonna do photosynthesis within the chloroplast. And the chloroplast has thylakoid membranes that are stacked up as grana, and the interior of the grana is called the lumen, and the outside is called the stroma. So I wanted you to have that, you could put right here. This is a thylakoid membrane, and across this thylakoid membrane is the electron transport chain. There are two electron transfer chains that you need to be comfortable with, and I'll walk you through that. But the purpose of the electron transport chain is to generate both ATP and reduced NADP. So when you do the light independent reaction, that is when you're reducing CO2 down into glucose. So let's take a closer look at the light dependent reaction. So here, you should always start kind of getting your bearings. So this is a phospholipid bilayer. These are the green membranes of the thylakoid membrane. Here's the interior of it, the lumen, and here's the outside, the stroma. We always want to start with water. Water is your source of electrons that you're going to ultimately use to reduce NADP. And these same electrons in NADP are going to be given to carbon dioxide to ultimately build sugar. Okay, put a pin in that, I'll come back to it. Right, so you need a source of electrons. You do that by breaking water in half. The oxygen is released as a gas, and the electrons are donated to the electron transport chain. The hydrogens from the water, the hydrogen ions, are going to be accumulating within the lumen in the interior of those thylakoid membranes. Like, think of them as hollow pancakes. These electrons are going to get excited by the sun to a higher energy level. And you're going to do a series of redox reactions through photosystem number 2. So the high energy electrons are going to go to an acceptor, who's going to release it and goes to another acceptor. One of those is a molecule called PQ. PQ has the ability to grab additional hydrogen ions from the stroma and bring them into the lumen. So now I'm starting to accumulate hydrogen ions to the interior. That creates a chemiosmotic gradient, which is like water behind the dam. It's a difference in concentration because I have more hydrogen ions in the lumen than outside. It's a difference in charge because they are positive ions, and it's a difference in pH. I'm lowering the pH of the lumen. And the reason why I want to do that is so that they can move with the concentration gradient out through ATP synthase complex. This is an enzymatic protein which will catalyze the reaction of forming ATP from ADP, an inorganic phosphate. This is critical because I need this energy, this ATP, to build sugar in the Calvin cycle or the light independent reaction. So just to review, the reason why we're building up these hydrogen ions, the source of them is from water, and also PQ bringing additional hydrogen ions from the stroma into the lumen. Okay, so that is photosystem 2. Photosystem 1, I excite the same 2 electrons I got from water, but this time the ride is shorter because I'm going to pass them to transfer them to NADP to reduce that NADP. Now NADP is carrying some high energy electrons that I can donate to CO2 in order to reduce it into sugar. Okay, that's helpful. All right. So this is the light dependent reaction. It has to proceed. Okay, the dark reaction, that's an old school name, or the light independent reaction, or the Calvin cycle. This Calvin cycle, when we start, we want to start with RUBP. RUBP, you can see it has 5 carbons in it. And just to demonstrate conservation of mass, we are going to say that we have 3 of them. So these are 3 C5 molecules with 5 carbons in them. That's preloaded already. You don't have to worry about that. We're going to bring in 3 carbon dioxide. Each one of those carbon dioxide has a single carbon, so we're going to say it's a C1 molecule, and we have 3 of them. we are going to snap on this CO2 onto the RUBP to give us a 6 carbon molecule. Again, 3. So we're gonna say we have 3C6. That process right there is called carbon fixation. That's the first step of this Calvin cycle. We have taken CO2 that is a gas and we have made it a solid. When you light it on here, it's very unstable. So each of these 6 carbon molecules breaks in half to C3 molecules. Not a lot of energy in them. So what we're gonna do is we're gonna take the ATP that we just made in the light dependent reaction, and we're gonna take some reduced NADP that we just made, and we're gonna use this energy in these electrons to take these from low energy to high energy P3P. Another name for that is PJF. That's kind of an old school name. So we have a total, we had 6, we still have 6. And these 63 are very high energy. In fact, so much energy, they are the equivalent of 1/2 of a glucose molecule. Now, we are just gonna take one of those 6 away, and that's represented right here, this 1 G3P. So we remove 1, 6 minus 1 is 5. Now we have 5 G3. Lots of high energy. What's the significance of this? I brought in 3 individual CO2s, think of them like free-range chickens, they're all over the place, they're gas. And I'm pulling out 3 carbons connect together that is a high energy half of a glucose. So 3 carbons in, 3 carbons out, each time I turn the Calvin cycle. All right, so I have 5 left. I want to keep doing this. So what I'm going to do to these 5 G3Ps is I'm going to regenerate my RUBP back. That's going to involve another investment of energy, ATP, so I can rearrange these 15 carbons in total back to another 15 carbons, right back to RUBP. I can then do this whole thing again. I'm going to do this Calvin cycle 2 times. Each time I do it, I'm going to invest 3 CO2s per turn. And each time I do the turn, I will pull out 1 G3C, which is a half a glucose. So if I do it twice, I will pull out 2 halves of glucose. So that's helpful.
Okay, now keep that in the back of your head.
I'm Miss.
Hi, Winnie. All right, so
Okay, I had a student just come in and needed a little help with photosynthesis, so I'm going to give you a quick overview. You ready? So there's the light dependent reactions and the light independent reactions. You need to do the light dependent reactions because they're going to give you what you need for the second one. So in the light dependent reaction, you're going to start with water. You're going to break that water in half, and as a result of breaking the water in half, you're going to have some oxygen, and it's going to give you electrons that you can get excited twice to create a chemiosmotic gradient to set you up to make some ATP and ultimately some reduced NADP. You will take both of those to your Calvin cycle, your light independent reaction, where in step 1, you will fix CO2 to RUBP. Then that's really unstable. It'll break in half. You're going to power it up with ATP and reduced NADP. And then you're going to have 6 halves of glucose. Peel 1 off, leave 5 behind, regenerate some more RUBP, and go again.
and now you're like, what? Okay. Yeah. All right. So this is about the level of understanding, what she just did, that you need for on level, for AP bio, high school bio, right? That's about the level that you need to understand. So we're going to kind of work through this. So here's what you're going to write. Okay. Get ready to start writing. All right. We're going to write this down. Write neatly, because you probably want to reference this later. I know for some of you that is damn near impossible, but try. Just try. Give it your best effort. All right. We're going to write this out first. N means a number, some number of CO3 plus some number of water, H2O, in the presence of sunlight is going to give you CH2O, some number of that, and some number of oxygen. And if you're not a good writer, don't worry, you can go back and look at this later. If you've got writing struggles, that's fine. And write it just like that with the little arrow and the sunshine. That's letting you know this is, some of this is shorthand and symbol, and you're doing that intentionally. The arrow is a symbol, the little sun is a symbol. You're going to draw some squiggly lines in a minute. Those are a symbol.
Yep.
Okay. So some number of carbon, oh, that should say, that's a 2, not a 3, people. I just realized that says 3. Make sure that says 2. That, this guy, this is a 2. Yeah, well, because I had 3. Oh, shoot. Take 1 second, fix that. Okay. Sorry. I just realized that was wrong. Okay, so some number of carbon dioxide combines with some number of water in the presence of light. You don't have to write this. That's what you just wrote. If you need to write it to remind yourself what it says, these are the words for the symbols you just wrote. Some number of carbon dioxide combines with some number of water in the presence of light energy to produce some number of carbohydrates and some number of oxygen. That's what you wrote. Some number of carbon dioxide combines with some number of water in the presence of light energy to produce some number of carbohydrate and some number of oxygen. All right, that's what little n means, is some number. And typically, this is going to be represented this way. You can write this out.
This is CO3 again.
Oh, yeah, because I copied it. Yeah, that's 2. I'm going to go back and fix that one. I did not realize it said that. Man. That's what happens when you copy and paste stuff and it gets goofy on you. Sorry about that. So yes, this is often how you're going to see the reaction for photosynthesis written, just like that. 6H2O, 6CO2 plus 6H2O in the presence of light yields C6H12O6 plus 6O2. And if you notice, you're in multiples, you're kind of dealing with multiples of 3 in a way. All right, whoop, did everybody have that?
Not yet.
Give you a second, make sure you write it. And in words, this would be 6 carbon dioxide plus 6 water in the presence of light yield 1 sugar, glucose, and 6 oxygen molecules, not atoms, 12 atoms, right? Okay, so now we're doing a light dependent reaction. So if you want to draw a little line, you can draw a little line in between. That was our overview, what we're doing at the top. Now we're going to break it down. This is the light dependent reaction portion. So literally write this: water, 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 H2O. Y'all are looking at me like you're crazy.
It's still light dependent reaction.
Yeah, this is all light dependent reaction. This is gonna make sense in a minute, I promise. Right now you're like, what the heck? Water, 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 H2O.
Would that be H?
Just,
Would that be 5H?
just watch. It's gonna make sense in a minute.
When I was working at summer camp last year, one
Mhm.
of our bosses kept repeating to us, water, water, H2O, dehydration, no, no, no.
Oh!
And we were like, yep, hydrating,
Drink, darn it, you're out in the sun.
hydrating.
The next thing you're gonna do is you're going to write this, and this represents that your water is broken down. And when you break down waters, you're not going to get oxygen by himself. He's going to find another oxygen. So what you're going to get, what it's telling you is, you're going to get free floating electrons that come from the hydrogen. And they're going to, ions, hydrogen ions, which is, you've got your E negative. E negative is an electron, for anybody who doesn't know. When you see E negative, that's an electron. And then you're going to get oxygen molecules. So write that out as best you can. Or don't, if you can't read anything you write. And then, what in the world? You're going to draw a sun, squiggly lines, and 3 dots, and then photons excite electrons. Photons are light. Light is weird. Light behaves as a wave and as a particle. When light behaves as a particle, it is a photon as opposed to a wave. Light's
Photo meaning
bizarre.
light.
Yes, photo meaning light. So sun, waves, and 3 dots. And then photons excite electrons. I promise it'll make sense in a minute. You'll see why. We're going to use Miss Sloan's awesome mnemonic. Or at least I'm going to offer it up to you. You can decide to use it or not. And then you're going to draw these little arrows. The arrows go into the photons, and then the arrow comes off the word photon and goes to these guys. You can circle photon if you want, if that helps, but you're showing that these little 3 dots represent photons, and electrons are right here, represented by the E negative. So you want to show somehow in your notes that photon, the word photon, are E negative. Now you're being sure that you recognize what you're talking about when you write it out. All right, now you're going to write PQ. If you want, you can write the whole word. That's it. Plastiquenol. PQ takes E negatives or electrons and makes them ATP, and it does it 3 times. See the 3? I'm going to take 3 electrons, sorry, and makes them ATP. Put the little 3 underneath so that you'll know that's 3 electrons. Hello.
Last talk.
Yeah, plastiquenol. You can just write PQ if you want. Takes electrons and makes some ATP. 3 of them. Everybody have that? We have it, we have it, we have it. All right. Draw a line. All right, now we're focusing right here on this little E negative that we just looked at. Okay, now what you want to do is you want to show, if you didn't already, that this is where your electrons are coming from. Some of you may have already circled. And then you want to show this underneath of it. Do you have a question?
So it's PQ takes electrons and
Takes electrons and makes them ATP. And then a 3 underneath. So NADP catches 2 electrons and reduces, which then becomes reduced NADP. Because remember, if you gain electrons, reduction involves gain. We've gained electrons, so we've now reduced NADP.
so write this below the.
Yeah, write that. NADP catches 2 electrons, or catches electrons 2 underneath. And that gives you reduced NADP. All right, now ATP plus reduced NADP, that's what the R is, leads to your dark RxN is an abbreviation for reaction, science and medicine, RxN, reaction. You can write reaction if you won't remember what RxN means. But dark reaction, or Calvin cycle, or light independent reaction. You can write it any of those 3 ways. Dark reaction, Calvin cycle, light independent reaction, whichever term you prefer. So ATP, yeah, flip it over. And ATP plus reduced NADP, yeah, goes to your Calvin cycle, light independent reaction, or dark reaction. Anybody panicking yet? Don't panic.
I was panicking the second you said, hold on for a second, it's gonna get like.
It will, all the stuff you're writing out will make sense to you in a minute. I promise. Right now you're like, why am I writing these symbols? Why am I doing these things? Yeah, it's kind of, it's like I'm playing a prank on, am I punking you? I'm not punking you. I promise I'm not punking you. All right, draw 3 big lines under there. So you know you're moving, now you're moving to something different. This whole time we've been under this light dependent reaction. So now we're moving to the
Dark.
Light independent, dark, whatever. All right, so remember where you wrote this? Reduced NADP? Like a line or two above? Remember where you wrote that? Okay, now write this. Keep this in your mind, just you're gonna go back to it in a minute. But you have C35 plus 3C5 plus 3C1 equals 3C6. That makes sense, right? Which is the same thing as 6C3. So what this is saying is if you have 3 5-carbon molecules and you add 3 1-carbon molecules, then you're going to get 3 6-carbon molecules, which can be rearranged into 6 3-carbon molecules because we have 18 carbon, we have 18 carbon. Yes.
Herbert Reigns is 18, 1 chemo.
No, we don't do those. They're too unstable. They're not a thing.
6.
Okay.
All right, now. There you go. ATP plus, go right back up to where you wrote reduce NADP. Circle it. You're taking ATP with that, and you're going to use it on this C6C3. So these together are going to work on this guy right here. You feel like you're drawing a treasure map yet? Circles, arrows. Flip it over, that's fine. Oh, you have it on the other side? Just draw your arrow however you need to show yourself where you're going. It's fine. Just, yeah, make your arrow go from one side to the other. You can do that. Then you're going to write, take away 1 PGAL. Now, the reason I wrote it like that is she's going to call it like PGAL or PGAL instead of PGAL. So when you hear AL, that's why. PGAL. And that is going to give us 3 G3P and 5C3. This is all what she just explained in the 7-minute video, and then she condensed it. This is that whole process step by step, but you're kind of writing it out symbolically. And then you're going to write, you use ATP to build 3C5 plus 3C1 equals 3C6 equals 6C3, which if you remember is what we did up here. And then you're going to write a big check mark at the end. And then you're going to draw a line. All right, now, remember where you had that before? Same thing. You had that before and you circled it, remember? So we have just done 6C3 check mark, right? Oh, look what's going to happen. Your reduced NADP from before is going to act on the 6C3 along with an ATP. So you're going to take your ATP and your reduced NADP and you're going to produce your 6C3. So this guy plus this guy. And the reason we're drawing circles and arrows is because I don't want you to think it's a different molecule coming out of nowhere. It's the same molecule. You're going back to it, right, within the process. So I don't want you to think it's like a different NADP. It's the same one we had before. And don't worry, if this is crazy and nutty and you can't understand it or can't read it later, you can do it later, or you can just watch the video that's going to follow all this where you'll go, oh, I understand why I drew all those arrows. All right, so then you're going to write, take 1 away, glucose equals 5C3. I love how about 2/3 of you are trying to keep up and the other 2/3 of you are like, I can't write anymore. Like, I don't understand what I'm writing. It doesn't make sense. That's okay. That's all right. You're not supposed to know what's going on yet. This is an exercise in note taking, but it will make sense.
on the way.
Yep. And then you're gonna use that ATP to build 3C5. Now we had 3C5 up here, remember? Okay, but you're gonna build another 3C5.
He's like, we already wrote that down.
Yeah. You did. Draw another line. Because it's some of this is cyclical. It loops itself back around. Mm-hmm. Another line and another line. Make sure you're real clear. 3 good lines. Dash, dash, dash. All right. Now, are you ready? Look over what you wrote for a minute.
Oh, yes, yes.
What's you right there?
All
What's you right?
right. Now, what you wrote out is a step-by-step symbolic representation of what you're about to see. And if you watch this and then look back at what you wrote and go back and forth a couple times, you'll see how you could take your notes and do this after you've kind of memorized this, how your notes can kind of cue you in. All right. This is the best mnemonic I've ever seen. All right. So pay attention. This is great.
So this is a photosynthesis song, and I got some brave people here to do our photosynthesis song together, so here we go, ready? Water, 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 water. What are we gonna do? Break it in half. What do we get? Oxygen and electrons. Sweet, sweet. Here comes the sun, traveling in waves. Particles of light are called photons, and it gets the electrons excited. You and to make some ATP. Here comes the sun, traveling in waves. Particles of light are called photons, and it gets the electrons excited. Who catches it? NADP, forming fruit juice, NADP. Then you take some ATP and so reduce NADP to the dark reaction, or the Calvin cycle, or the light independent reaction. Here we go. 3C5 plus 3C1 makes 3C6. Break down to 6C3. Use some ATP, use some reduced NADP on your 6C3. Take one away, PJL, D3D. And you're left with 5C3. Use some ATP to build 3C5. 3C1 is 3C6. Break down to 6C3. Use an ATP.
Right.
Use a reduced NADP on your 6C3. Take one away, glucose. And you're left with 5C3. Use some ATP to build 3C5. Now, if that song is complicated at all, watch the other TikToks where I explain it. Okay? Hope you have a
So
great
this time watch it again, but look at your notes. Okay, so like watch what she's saying, but look at the notes you wrote and see if you can understand what it's showing you. Okay?
The girl with the second.
Yeah?
Oh.
Hey, ready? Okay, watch your notes.
Wow.
Hang on.
Gather
Follow your notes while she talks.
Water, 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 water. What are we gonna do? Break it in half. What do we get? Oxygen and electrons. Sweet, sweet. Here comes the sun, traveling in waves. Particles of light are called photons, and it gets the electrons excited. PQ, and to make some ATP. Here comes the sun, traveling in waves. Particles of light are called photons, and it gets the electrons excited. Who catches it? NADP, forming fruit juice, NADP. Then you take some ATP and so reduce NADP to the dark reaction, or the Calvin cycle, or the light independent reaction. Here we go. 3C5 plus 3C1 makes 3C6. Break down to 6C3. Use some ATP, use some reduced NADP on your 6C3. Take one away, PJL, D3D. And
I
you're
love
left
that.
with 5C 3C3. Use some ATP to build 3C5.
3C1, 3C6.
Break down to 6C3. Use some ATP. Use a reduced NADP on your 6C3. Take one away, glucose. And you're left with 5C3. Use some ATP to build 3C5. Now, if that song is complicated at all, watch the other TikToks where I explain it.
You see what you wrote?
Right there.
Do you see how what you wrote is what she said? So you can listen to her while you're watching it. And then once you kind of get the cadence, the hand motions may look goofy, but I'm telling you, there are enough scientific studies that show that when you take physical movement and connect it with a mnemonic device like that, you remember it better. Uh-huh.
I heard people whispering jazz hands. I would do that.
Jazz hands. Yeah. Uh-huh.
I love how they like they say it at the same time and then
Yeah.
look how much tries they did.
Oh, they probably practiced the heck out of this to get to this point. But it's catchy and you remember it and it makes sense. She has we're gonna see a fair amount of her this year. Probably not as much as we see of Professor Dave in chemistry, but a fair amount of her because she has a ton of these and they're great. They're really good. So I recommend if there's something that we're going over and you're like, I don't know if I get it, check out Miss Sloan's thing and see if she has it because she very well might. And it's super helpful. So you've got something there to work with. We're going to do it one more time and I want to see how many of you can follow along with at least what she's saying. If you don't do the hand movement, see if you can follow what she's saying and copy.
Okay.
Oh, no, Miss Sloan, we got to go back. Go back, Miss Sloan. All right.
This is a photosynthesis song. They got some great people.
Which is
to do our photosynthesis
great
song
memory
together.
device.
So here we go. Ready?
Water, 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 water.
What are we gonna do? Break it in half. What do we get? Oxygen and electrons. Sweet, sweet. Here comes the sun.
Traveling in waves, particles of light are called photons.
Let's see
Electrons excited. What's this? Electron chain, right?
You. Make some ATP.
ATP.
Here comes the sun.
Traveling in waves, particles of light are called photons. It gets the electrons excited. Na 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 na.
Catch it. ATP. Form reduce
ATP.
Then you
You
take
take
some
it ATP and reduce NADP
to
to
the
the
dark
dark reaction.
or
Or
the
the
power
Calvin cycle, or the light independent reaction.
Yeah. 3C5 plus
3C1
is
3C6
Break down to 6.
6C3.
Use some ATP. Use some reduced
Reduce NADP
on the 6C3.
6C3.
Take one away.
Away.
PJL. G. Breathe. And you're left with 5C3. Use some ATP to build 3C5.
3C1 equals 3C6.
Break down to 6.
6C3.
Use some
ATP
Use
to reduce NADP
on your 6C3.
6C3.
Take
One
one
away.
glucose. And you're left with
5C3.
Use some
ATP
to build 3C5. Now, if that song is complicated.
There you go. So some of you actually followed along. That's cool. That's cool. It's really great. She's got great devices. All right. So do not show up at my door on Tuesday. You will probably not like the way I look because I will probably be in my pajamas and that will be scary for most of you. So don't do that. And enjoy your fall break. All you have is normal homework, no lab right up. So enjoy yourselves. Get outside, cut the grass, all that good stuff.
Getting it back.
There, you answered something. I know you did.
No. 20. No problem. You like.
All right, there we go.
How often do you actually?
What's the matter?
He just wrote a piece of paper that says Barrett is gay like 20 times. And now he wants it back because it took him time to do.
Oh,
I'll throw it in crap.
nor is it an insult, so. Shall I just say?
Oh.
Oh, my.
Yeah, you look tired. I'm gonna go home and read all of them too.
Oh my goodness. You did knock me down with the feather when I learned your brother was joining a fraternity.
These aren't, it's crazy.
I know, that's the craziest thing ever.
You knocked me down with a feather when he said he didn't want to join one. I was like, yes, you do.
I
You
didn't
want
figure
to be
he
in
would.
the
I was like, I don't know, Cole never did, I didn't think Michael would, and then when Cole talked to mom last week and she's like, you'll never believe it. Cole said Michael's driving a frat. I'm like, oh, and then last night he sent Jason and I the picture of
Yeah.
Michael with his frat bros, and he's like, look real close at this picture, you won't believe it. And we're all like, no, we don't believe it, that's craziness.
It was the one my grandpa was in.
Oh.
He only joined it because they promised that if you said no to anything, that meant no.
That, no, yes.
That's so he's like, okay, I'll do this.
Yes, yes. Such cool. Good for him.
He came home for the weekend, but we were on retreat.
Did he come?
So we saw him for like 30 minutes or something before we left again.
Nice though to see somebody I never see mine. So
Never comes home.
yeah, no, we had him for all 3 weeks and 3.5 weeks or whatever this summer. We were
Yeah.
like, oh, we're so special. But we're going in well when James is staying here, Caitlin's brother's gonna stay here with Ozzy. Michael's gone. He was our last Ozzy keeper. But we're gonna go out there for Halloween weekend, basically the weekend of the first, and yeah, I'm gonna take my mom because she's never been, and we haven't been in 2 years, so
Yeah, since you moved him in, right?
we went for family weekend after that, but that was it.
Yeah, that's where we'll be.
I mean, we've been to Dallas because Drew has a gym that's in Dallas that we could go to, but we haven't been to, like, we haven't seen his house or whatever. He's already
Oh,
asking.
that's right. He does.
Yeah, he's got a house this year, so he was asking if my mom and I could make dinner for everybody, and we're like, we knew that was coming. Of course we will. Like, we knew that would be a thing. So yeah, it's gonna be
That's
fun.
awesome.
Have a good time, and James will be here with Ozzy, so Ozzy will be fine because there's only a handful of people he actually likes. So I was like, who can I ask? I can ask James. I can ask Colin. Oh, it's that cool way. You wanna come, Mommy? Oh, did you see what he did yesterday? He dug 2 giant holes in the yard yesterday. Oh, we found something to share. I don't know if it was a shit or this one, but this one we got. And one of the holes was massive, like, doing the docks and just came on. It got way bigger push. I put it in holes and full on docks today. You're what? Oh, this is what we did. Such a jerk. I was like, yeah. I went outside because it was dark. So we talked about it. And I brought it in, and then he did this idiot stuff. This. Look, we're gonna step right along. What is wrong with you? And then I had to look at his nose, right up in here. Oh my gosh, for real. You're the best. That's it. You would. Your best thing. Oh, yeah. It's funny because it's like the people he knows and loves, he knows he's. The people he doesn't know yet, he's like, I don't know about him. He's gonna what we're excited about. Yeah, he's a mama's boy, he does not sleep with us when Jason's home. But he will sleep with me when Jason's not here, and he knows, he's figured it out by now. Like people look at me when Jason's not here, and he'll look straight, look at me, and then he'll go upstairs, like, oh, I didn't sleep with you, but we don't even have to, like, at night, we tell him to go out, we're going up. But now, like, literally telling myself, for the television off, he's like, oh, that's time to go to bed, so he'll take himself out, and he'll go right to straight. Tuesday mornings, he goes right to his right. He knows that he's doing it, so he goes right to his right. You got a little bit of long tone, and shouts. Oh, Stanford, so that was huge. Yeah. So I hear about it in Senate for certain. People can't excited enough. It's like, dude, why? Yeah. You don't like it all, really. Not funny bad. You're right, just be he's so soft. He's so soft. Real.
Wait, and time to stop those.
Yeah, like, excuse me, I ordered an infection, the infection's stopped. I know, I love you. You look on the floor. Yeah, that's what I'm talking about. Aaron, you know.
Dr. Muddy. He has to.
One of my friends in elementary school, he and his family outside the house, they was put on the road and all over the week. He's probably got, he's not it. I don't know about it. And he's slow, yeah, well, this, they were there. Well, he does, he does, oh, it's more. We're gonna see what's up there. There's the, oh, and, all I need is, here are the things. Next month, seven. No, 6. Or 6. 29. November 20th. Everly, he nips Everly. And she gave us a surprise sneak attack hug and he freaked out. Good boy. There's no nice good boy. He did some, he knows, some furniture's changed and the chairs, things are out, whatever, and he goes upstairs with me. And then he will come down with me and he'll write this grade. And he just goes like, let's do this, great, just goes right this grade. That's funny.
Sometimes like when you go, there's one going to take me somewhere, so like we should get taking there, I'm going to like, and this or that.
He actually,
Holidays and meeting around the kids,
last week, he barked at Jackie all the way downstairs. And she's like, huh? Came over and he looked at her and like gave her a hello and then went straight to straight. She's like, he just put himself in his room. Hey. Come on. Come on.
Let's be going, yes, you know.
Yes, you are.
Bye,
Bye, guys. Have a nice break. Oh, good boy. Oh, good boy. And then turn off the Zoom. I always forget to turn off the Zoom.
Alright, let me mom.
Hold on. Gotta end the Zoom.
Buddy.
Maybe. Come on. There you go. All right, you going to practice?
Yep.
Love you. Have a good one.